Good morning, friends and students. We are in B.Sc Zoology paper 2 Mammalian Histology class. Today we have a new topic, epithelial tissue. Epithelial tissue are distributed all over in our body, especially on outer lining of our skin, inside and outside. In this photograph you can see various location of epithelial tissue in our body. A simple squamous epithelial tissue, simple cuboidal epithelial tissue, simple columnar epithelial tissue, stratified squamous, stratified cuboidal and stratified columnar. These are the different types of epithelial tissue you see distributed in our body. Always remember, these epithelial tissue are all the times either forming the internal lining or external lining. For example, our skin is all epithelial tissue. Internal lining of blood vessel is all epithelial tissue. And this squamous, cuboidal, columnar, this is terminology based on their different functioning, and you see there is a difference in their size also, as if you compare it with the squamous, cuboidal and columnar. We'll see in detail every types further. So epithelium consists of a vascular sheet of cells which cover or line exterior surfaces of body, closed cavity from inside and outside of that cavity, that is cardiac cavity, thoracic cavity and abdominal. These are the different three cavities you find in our body. Tubes which communicate with the exterior, such as alimentary canal. Again here the internal lining of alimentary canal is made up of epithelial tissue as well as external lining. Respiratory tract, all the tubes, internal lining of tubes in respiratory systems, they are all epithelial tissues. Then genitourinary tract, that means reproductive system, tubes in reproductive system and urinary systems, they all are internal outer cells are of the tissue, they are epithelial tissues. What are the functions of epithelium? They serve as impervious barrier. Barrier means which arrest transfer of something. Here they arrest and they selectively allow passage of some electrolytes or water. So they serve as a barrier that you can see in epidermis. If we immerse our hand in water, that water don't get percolated in our body. So that epidermis arrests the entry of excess water in our body. Urinary bladder, where urine get concentrated and remain there only, it don't get again absorbed in our body. That way the barrier works. Secretion, exocrine glands. Second function is secretion. Here all exocrine glands, they are epithelial tissues. They serve to secrete some secretions. So everywhere, like salivary gland, there is a secretion of saliva. The saliva is secreted from epithelial tissues. Absorptive, internal lining of intestine is all, it is a epithelial tissue where they absorb nutrients from our digested food. So you will find that absorptive type of epithelium in an intestinal mucosa. Fourth function of epithelial tissue is the transport. Now transportation doesn't means that transportation of some molecules, but here the concern is motile cilia. In internal tracheal lining, we have ciliary epithelial tissues. So if any dust particle enter in our trachea or nasal passage, that get removed by continuous ciliary movement of that cilia. which remove that particle and arrest entry of that particle in our respiratory system. Fifth function of epithelium, you find that is a sensory. So in our body, that photoreceptor cells or auditory cells, they are primarily epithelial tissues. We see pictures, we see vision is only because of a epithelial tissue. They serve as a photoreceptor, and in auditory cells in ear, where there is a cilia-like structure, they receive the waves, sound waves. So sensory structure, even in our skin there are some sensory cells, sensory tissues. So sensory is one of the function of epithelium. Now epithelial tissue, we classify that epithelial tissue. The classification is entirely based on their structure. Structure, simple one layer of cells, that is simple epithelial tissue. Stratified, where there is a several layers of cells, forms, that is stratified epithelial tissue. And on the basis of shape of a cell, that is squamous, cuboidal and columnar. In case of squamous, that shape is very flat, like a page of your notebook. Cuboidal, where the shape of epithelial tissue is a square, cube-like. And columnar, where the shape of that epithelial cell is elongated like a column. So overall, here we have seen where we find epithelial tissue, what are the functions of epithelial tissue, and how this classification of epithelial tissue is based. Now in next we'll see this different simple, stratified and squamous, cuboidal, columnar epithelial tissues in detail. So here in single picture we can see, or we can show different types of epithelial tissue at a glance. When we say a squamous epithelial tissue, they are very flat. In every cell you will find a nucleus, a bulging nucleus, a round shape. And one more point we must take consent of is the basement membrane. All these epithelial tissues are settled on some strong base, which is non-cellular, non-living, and it is called as a basement membrane. You will not find any nucleus in that basement membrane, so it is a non-living. So squamous cells are settled over this basement membrane. You can see they are hexagonal shaped cells, but they are very flat. They are so flat that you can see the bulging of nucleus on these cells. Thereafter, cuboidal cells. So here you can see these cuboidal cells. The shape of that cell is like a cube, and they are all arranged in one layer. Here also squamous cells are arranged in one layer. These cuboidal cells, I hope you understood and you will remember this name of cuboidal epithelial cell on the basis of a cube. We'll see actual photograph in next few slides. There you will understand more. Now columnar epithelial cells, they are very elongated and long like a column. Nucleus is also there, but the length of height of a cell is very long, so called as a columnar cells. This is a pictorial diagrammatic view of different type of epithelial tissue at once. Now here in your notebook or in exam, in your notes, you can draw this diagram. While writing explanation about this epithelial tissue, you can use this diagram showing all these three simple epithelial tissue, squamous, cuboidal and columnar over the basement membrane. Now simple squamous epithelial tissue. It's very thin and paper-like structure with small bulging nucleus. You can observe. Where could you find this simple squamous epithelial tissue? On internal lining of blood vessels. That is, there they are called as a endothelium. Though the name is different, the basic name is simple squamous epithelial tissue. Now here actual line drawing of actual cells or actual tissues of this simple columnar epithelium. You can easily note a column-like structure of this epithelial tissue. Here they have shown column-like epithelial tissue but with cilia. So ciliary columnar epithelial tissue. In this case, these are the simple columnar epithelial tissue. And thereafter this simple cuboidal epithelium, you can see a flat arrangement of cubes over the basement membrane. So simple cuboidal epithelium. You can compare this simple squamous epithelium with the simple cuboidal epithelium. Why simple? Because they are all arranged in a single row, flat structure. Here again you can see they are all arranged in a single row and flat in structure, but shape is cuboidal. So cuboidal epithelial, simple cuboidal epithelial. If you able to understood these basic different types of epithelial tissue, it's very easy to learn the histology, mammalian histology, because all these names, terminology is important in understanding further different organs histology. Now here they have given a color actual photographs of tissue from the mammal rat. So how this simple squamous epithelium look like in actual section. So here you can see this uppermost lining, a black spot here you can observe a dark spot. These are the nucleus and surrounding is a cytoplasm. So this is a one single cell. These are all single cell lining. So it is a simple squamous epithelial lining a tissue over the skin. This is a skin section. Thereafter, if you want to see a cuboidal epithelium in actual tissue, here you can see this is a one cell. This is a one cell, a cube-like structure, a large nucleus and surrounding cytoplasm, and it gives a shape of a cube. So this one cube, second, third, fourth, so all these cubes are lined in a single row, so called as a simple cuboidal epithelium over this basement membrane. This is a simple columnar epithelium. Now simple means they are in single row. They are not overlapping, so it is a simple. Columnar because they are in a column fashion. Here one thing you will notice that all these nucleus are at the base. They are located at the base and above portion is filled with the cytoplasm. So this is called simple columnar epithelial tissue. I hope it required little bit imagination to visualize all these different types of theoretical tissue structure in actual tissues. Every time I will try to explain this thing in actual tissue, how this cells arrangement is there and how that cell looks and why they are named like that. Now in studying histology, it is not possible to see all the histological structure in three dimensions, all the histological structure in three-dimensional view. So while making the slide to observe under microscope, that tissue may get cut into different planes. So you will find in a syllabus different label like skin BS, tooth LS, intestine TS. What this LS TS word stand for, that we'll discuss here in this slide. The plane of sectioning. Sectioning is when we cut that tissue into thinnest possible section. When we cut that tissue, it may goes in different plane, and accordingly we see the structure under microscope. So before cutting any tissue, we must know in which plane we are cutting that tissue. So here, if suppose this is a blood vessel, a column, round column like blood vessel is there, and we want to see the transfer section of blood vessels. We want to see the different arrangement of cells and tissue in blood vessel. We must cut that blood vessel in a transfer section. So it is a transfer section, and now we abbreviate it as a, this is a transfer section and we abbreviate it as TS. If we want to see some different, if we want to see this in oblique fashion, we cut this in a oblique way, oblique plane. And longitudinal section, if we cut this blood vessel in this two half longitudinally, it is a called longitudinal section. Now what happen when you see actual slide photograph, color photograph, why you did not able to understand these epithelial layers in any actual tissue? The reason is, see how this tissue get cut. Here every time while taking a section of tissue, we must know in which plane we are cutting and label that, so it will help us to imagine this arrangement of that tissue in that section. Here simple squamous epithelium. This is again a diagrammatic view of simple squamous epithelium. All the cells are in close contact with each other. Centrally there is a nucleus located and cytoplasm bulging, and they are very flat. This is how they look actual under scanning electron micrograph. Simple squamous epithelium. All this bulging is because of nucleus, and these are the boundary of that cell. So entire this sheet of cell is called as a simple squamous epithelium. Again we go back to previous slide, and diagrammatic presentation of simple squamous epithelium comes like this. Same arrangement of hexagon with bulging cytoplasm and nucleus, which you can verify with this scanning electron micrograph of squamous epithelium. A simple columnar epithelium, how they look like. Here why we call them as a column, because they look like this when you see them under electron micrograph. And here these are the boundaries of a single cell. Simple columnar epithelium. Now here different views of columnar epithelium they have given here, and these are all scanning electron micrograph to confirm our understanding. This is a basement membrane, and these are all columnar cells lying with each other in a single layer. Here again a magnified views of that is a basement membrane with this columnar like cells arranged. Lateral section, when they take lateral section you can see like this. If you crack that section, that layer, this is how the cells get separated, and you can confirm that columnar, simple columnar structure. But here this whitish lining indicates a cilia. It is also called as a brush border lining over the columnar epithelium. So now they are termed if you find such ciliated columnar, simple ciliated columnar epithelium. Here again in light micrograph, a simple columnar epithelium look like this. So in regular college compound microscope, you can see this columnar epithelial tissue like this. Here it is a nucleus darkly stained, and this is the entire cell portion, and this is a brush border, that is cilia. So ciliary simple columnar epithelial. This is generally lining of intestine, internal lining of intestine. In between somewhere you find such glands, goblet cells. We'll discuss that later on. They are focused to the columnar epithelium here only. With different stain, these epithelial tissue can be visualized in different ways. Simple epithelial lining tubes. Here in simple cuboidal epithelial lining, convoluted tubules in kidney. So this is a section of a kidney. Here all these are the tubules. Seminiferous, this is a section of kidney. Here you can see all tubules cut in a section, transverse section. These are all tubules of nephron, single functional unit of a kidney. When they cut in different way at different locations, you see the increasing and reducing diameter of these tubules, uriniferous tubules. If closely you observe, all these cells looks like a cube, arranged centrally pointed, and have central lumen. Every these cuboidal cells have nucleus inside. So this is how we'll see these cuboidal epithelial in different organs in our mammalian tissues. Here they have shown a convoluted tubules cross section. In case of simple squamous epithelium, the endothelium lining the lumen of small blood vessels. If you cut blood vessel in transverse sections, so this is the blood vessels here. In this blood vessel, all these black dot indicates the nucleus of a simple epithelial lining tube. Okay. So every nucleus means single cells. Every dot black spot indicates the single epithelial cell, and so this entire lining is of a simple squamous epithelial tissue, commonly called as the endothelium. Here simple columnar epithelium lining papillary duct in the kidney. This papillary duct functions like to absorb some electrolyte while forming the urine. So in LS section, longitudinal section, if you cut that duct, you will see all these single duct cut into the longitudinal section. So we covered that different sectioning slide earlier. Now here you can see a specific columnar arrangement of cells. They indicate absorptive nature. All these tubules are internally lined by column shaped single layer epithelial tissue, so called as simple columnar epithelial tissue. Now another way of, now this is next level of epithelial tissue. Earlier we have seen simple. Simple. We everywhere use a simple word for classifying these epithelial tissue. Simple squamous epithelium, simple columnar epithelium, simple cuboidal epithelium. Now here in next level you will find they have used a stratified word. Stratified means when there is a layer by layer arrangement of a epithelial tissue. So that may be of simple epithelium, that could be a columnar epithelial tissue or pseudostratified columnar epithelial tissue. So different three basic type of epithelial tissue when arranged in layers, they are termed as a stratified epithelial tissue. In case of this stratified squamous epithelium we understood earlier, squamous means flat. We understood earlier squamous epithelium means flat. Now stratified means many flat squamous epithelial layers are arranged on each other. So this kind of arrangement is now termed as a stratified squamous epithelium. It's very easy to understand this terminology. A many layer of squamous epithelium, that is stratified. So here again many layer of columnar epithelium arranged on each other, that is stratified columnar epithelium. Pseudostratified columnar epithelium. Now here, though they look like they are arranged on each other in a layer, but they are not. They are single layer. So named as a pseudostratified columnar epithelium. They are simple columnar epithelium. Simple arrangement, no overlapping. Just one more type is a transitional epithelium, where if that tissue or organ is in collapsed form, you will find all the epithelial tissue layers, they are arranged on each other. So it looks like they are stratified tissue, stratified epithelium in a collapse, that is relaxed form. But when you stretch that tissue, it becomes flat, a single layer. When they distend and when they collapse, they form a layers, and when they distend, they form a single layer. This generally happen in case of a urinary bladder. When our urinary bladder is empty, if you take section of an empty urinary bladder, you will find such kind of arrangement of epithelial tissues. So it doesn't mean that they are stratified squamous epithelium, but when you stretch these epithelium extensively, when the bladder filled with urine, it gets stretched and they become flattened. In that case, if you take a section, it look like a single layer. So these kind of epithelial tissue which shows a dual nature, they are called as a transitional epithelium. Sometimes it is flat and it can transit into another form as a transitional epithelium.